Hello friends and dear students. Today in continuation with a topic biomolecules, I am going to discuss about the chemical structure of disaccharides. So you know disaccharide, that is maltose. So maltose, it is made up of glucose and glucose. If you see, there is a chemical bond between these two units of glucose. So what type of chemical reactions occur between these two units of glucose? Here you see that is O linkage, that is known as glycosidic bond. What happens if you see the structure like this glycosidic bond, then you will find there is a OH at carbon number 1, and then you will find OH in the second glucose molecule at carbon number 4. So this OH and OH, there is a formation of one water molecule with the help of this hydrogen and this OH. So there is a release of one molecule of water. So if you see chemically, then we can say this glycosidic bond is alpha 1,4 linkage. What do you mean by alpha 1,4 linkage? Means this glucose is alpha forms, alpha D glucose. It means hydroxy group is present below the plane. Suppose this is the plane, so at the hanging position, like if you say this is hydroxy group, so it is present below the plane of these ring structures. And now, if you see what type of this glucose it is, you know, this is beta D glucose. Why I am telling beta D? You have to recognize at carbon number 1, you will find hydroxy group above the plane of the ring structure of this glucose. So there is a reaction between the hydroxy group present at the carbon number 1 of the first glucose unit and hydroxy group present at carbon number 4 of the second glucose unit, and then it will release one molecule of water, and this oxygen will create a linkage that is known as glycosidic linkage. So this is the disaccharides, means two units of glucose joined together by a glycosidic bond. Now I am going to discuss the next disaccharide, that is lactose. The maltose is generally present in malt, that is barley, and lactose generally present in milk. So in so many competitive exams, generally ask that give two example of disaccharides and what is the monomeric unit. In case of maltose, monomeric unit is glucose and glucose. But you have to clarify what is the form of this glucose. This is alpha D glucose and this is beta D. Okay. So we can say it is alpha 1,4 linkage. That is the glycosidic bond between two units of this glucose. But if we see the lactose, that is milk sugar, it is made up of glucose and galactose. How can we differentiate glucose and galactose. So if you see the open chain structure, this is glucose and this is mannose and this is galactose. So I am concerned with the chemical structure of glucose and galactose. If you see at carbon number 3, hydroxy group present at the left side in open chain. In the same way, in case of galactose, at carbon number 3 and 4, hydroxy group present left side. So if hydroxy group present left side, then in ring structure it will evolve the planes. Okay, so you have to know how this bond will form between glucose unit and galactose unit. Okay, so if you see, in case of galactose, suppose if you want to show, this is hydroxy group OH and this is H. Okay, and then you have to make a bond between alpha D glucose and alpha D beta D galactose. This is, suppose this is alpha D glucose and this is beta D galactose. So generally feeling confused why it is known as alpha D glucose and why it is known as beta D glucose. Why I am writing this OH? Because this at the carbon number 4 hydroxy group left side, so you have to show OH above the plane, and in this case it is alpha D glucose. So a chemical reaction occurs between this two hydroxy group, means carbon number 1 of the glucose unit hydroxy group and carbon number 4 of the galactose unit hydroxy group, and it will make a water molecules and then release of H2O after chemical reactions. So you can show it is in this form, that is bonding, you can show like this form. Okay, why I am telling that you have to show this bonding in this form? Because in one case hydroxy group in glucose is below the planes and in case of galactose it is above the planes. So you have to show like that. Okay, in the same way, if we observe sucrose, okay, then in case of sucrose, one unit of this disaccharides is glucose and one unit of is fructose. So try to differentiate. This is aldehydic sugar and this one is ketonic sugar. So there is a chemical bond formed between carbon number 1 hydroxy group and carbon number 2 hydroxy group. Why? Because this is ketonic sugar. So you have to make a glycosidic bond between alpha 1 and 2. What do you mean by alpha 1 and 2? Means the carbon number 1 hydroxy group make a chemical bond with hydroxy group present at the carbon number 2, and then it will make a glycosidic bonds. So you have to understand why I am writing this. In case of fructose, this is anomeric carbon, anomeric carbon in ring structure. In case of glucose, this is anomeric means first carbon in case of ring. So this ring is pyranose and that ring is furanose. Furanose means F means furanose and F means 5. So 5FF. Remember like that. So that is 5R ring structure. And you will find oxygen means. In case it is 6R ring structures. So that's why we are indicating that the linkage between this 1 and 2. In case of sucrose, both the anomeric carbon hydroxy group involved in chemical bond formations like glycosidic bond formation. That's why there is no free hydroxy group present at the carbon number 2. And anomeric group, hydroxy group present at anomeric carbon is responsible for chemical reactions. Then we can say it is reducing sugar because this hydroxy group react with copper sulfate, salt of copper, that is in phalanx and binding solutions, and initially it will give yellow color and further red colors. If it is giving red color, then we can say it is reducing sugar. If it will not give red colors, then we can say it is non-reducing sugar. Means if there is a presence of free hydroxy at carbon number 1 of any 2 units, then we can say it is reducing sugar because this reducing sugar react with phalanx reagents or basic solutions to give yellow and further red precipitate, just to estimation of sugar in your bloods in any chemical level. Okay. Now, question will ask, which of the following are reducing sugar? You will get the choice maltose, like lactose, and all monosaccharides. Means all monosaccharides are reducing sugars. Disaccharides, in case of disaccharides, lactose is reducing sugar and maltose is reducing sugar. Then question will ask like that, which of the following is not a reducing sugar? And you will get a choice like sucrose, lactose, maltose. Then sucrose is non-reducing sugar because the anomeric carbon hydroxy group of both the monomeric unit involved in chemical bond formation, that is glycosidic. That's why we can say sucrose is non-reducing sugar. Okay. Now, one word in previous class I have discussed epimer. What do you mean by epimer? Suppose this is the straight chain structure of glucose, this is the straight chain structure of maltose, and this is the straight chain structure of galactose. If you compare one by one with glucose, then you will find at every point, suppose I am taking this carbon number 3 in case of glucose and galactose, and rest other, you will find the similar structure is present. Position of hydroxy group, position of hydrogen, and aldehyde group already present at carbon number 1 in case of glucose and mannose. But if you see at carbon number 2, there is a difference in position of hydroxy group. In case of mannose, hydroxy group at carbon number 2 present at left side In case of glucose, hydroxy group present on the right side at carbon number 4. So we can say these two are epimer. If two chemical structure, that is monosaccharide, differ at only one carbon due to position of hydroxy group, then we can say it is epimer. So glucose and mannose both are epimer to each other. Next, if you compare this glucose with galactose, this is the chemical structure of galactose and this is the chemical structure of glucose in open chain. Then if you find at carbon number 4, what is the difference? You will find in case of glucose, hydroxy group present and the carbon number 4 is right side, but in case of galactose, hydroxy group present on the left side. Only this carbon position of hydroxy group is different. That's why we can say glucose and galactose are epimer. Means single point difference due to position of hydroxy group or hydrogen in any carbon of the two monosaccharides, then these are epimer to each other. But you cannot say mannose and galactose are epimer because they are no differ at only one carbon position in a straight chain. So we can say it is not epimer. Now come to find out what is chiral carbon. It is very important in organic chemistry or biochemistry. Means just to find out isomers of any biomolecule, especially monosaccharides. Okay. So if you see this carbon, then we can find this carbon is surrounded by 4 different groups. Like we can treat as this is the separate groups. So we can say this is chiral carbon. If any carbon surrounded by 4 different groups, then we can say chiral carbon or asymmetrical carbon. Okay? But if you observe at carbon number 6 in any monosaccharide like this one, if you see here, there is one hydrogen and one hydrogen and one hydroxy group. So these two groups are similar. If the carbon is surrounded by similar type of groups, then we cannot say it is chiral carbon. It is a chiral carbon, means symmetrical carbon. So by counting the number of chiral carbons, we can say the possible number of isomers. We cannot say this is chiral carbon, it is not a chiral carbon, but if you observe minutely, then it is also a chiral carbon, like it is a disaccharide group. So in case of glucose, there is a 4 chiral carbons. Like this is, suppose this is the structure of glucose, this is, there is 4 chiral carbons, 1, 2, 3, 4. If you observe this, we cannot say it is chiral carbon because there are no different groups surrounding this carbon. So we cannot say carbon number 1 is chiral carbon or carbon number 6 is chiral carbon. After counting the number of chiral carbons, you can find out possible number of isomers. Like 2 power n. If there is 4 chiral carbon, so you will get 16 isomers. So this is basic concept in case of organic chemistry. In case of biochemistry, you have to find out the chiral carbon, and then we can calculate possible number of isomers. Such type of question may be asked in any competitive exam or in any CBSE based exams. So this is our about epimers, chiral carbons, and then calculation of isomers, and then alpha D glucose, beta D glucose. Why it is alpha D? Why it is beta D? If hydroxy group above the plane, then it is beta D glucose. Okay. If hydroxy group above the plane, then it is beta D glucose. If hydroxy group below the plane, then we can say it is alpha D glucose. Alpha D glucose. So a question is asked frequently like, which of the following are epimers to each other? Mannose and glucose. One choice. Mannose and galactose, also epimer. But if there will be a choice like mannose and galactose, then it is not epimer. So you have to draw the chemical structure. I have already discussed the chemical structure of monosaccharides. Simply you learn some points and then quickly derive the structures. And what do you mean by pyranose ring? Means if it has 6 R ring, then it is pyranose ring. If there is a 5 R ring, then it is furanose. You know, carbohydrate is aldehydic or ketonic form of polyhydroxy alcohol. So why I am telling aldehydic? Means at every monosaccharides at carbon number 1 there must be aldehyde group present. Or if it is ketonic sugar, then at carbon number 2 ketone group will present. Besides that, there must be hydroxy group, alcohol group, and then hydrogen. So you have to judge whether the valency of carbon is fulfilled by in the chemical structure or not, or valency of oxygen fulfilled, because the valency of oxygen is 2 and valency of hydrogen is 1. So if you remember the valency, then you can draw the chemical structure of all the monosaccharides, chemical structure of all the disaccharides. Such type of question generally asked in any competitive exam. Anyone tell what is maltose and what is mannose? Mannose is monosaccharides, maltose is disaccharides. So you have to remember such type of things. Disaccharides means two monomeric unit joined by glycosidic bond. Monosaccharides means single unit, simplest form of carbohydrate like glyceraldehyde, erythrose, threose, ribose, arabinose, xylose, lyose, and next allose, altrose, glucose, mannose, glucose, galactose, iodose, tallose. These all are monosaccharides. Okay, so just with the help of a simple chemicals, Helling's reagent or Bandic solution, cupric oxide, it will give a reaction. Generally, cupric oxide blue color, but when it will react with monosaccharides, you can observe that is initially you will observe the yellow color and finally it will turn into deep red. Just for testing of any blood. So all monosaccharides, in case of disaccharides, maltose, lactose, these are reducing sugar. But sucrose is not a reducing sugar because anomeric carbon hydroxy group in both the units involved in chemical bond formation. So it will not react with Felix reagent or Van-Dyck solutions. That's why we cannot say sucrose is reducing sugar. And all polysaccharides is also non-reducing sugar, like starch, glycogen. Starch generally present in plants, and glycogen generally present in animals, muscles, as a storage food, carbohydrate, that is polysaccharides. Poly means many unit of monosaccharide joined together by a glycosidic bond. So that is polysaccharides. Oligo means few. If there is 3 unit of glucose joined together, then trisaccharides. If there is 4 unit of glucose joined together, then it is tetrasaccharides. If there is a 5 unit of glucose joined together, then pentasaccharides. 6 unit of glucose joined together, then hexasaccharides. So oligo means 2, 2, 6 or 7. Then we can say it is oligo. And monosaccharide means single unit, like glucose, galactose, ribose, arabinose, fructose. So these are monosaccharides. But there is a chain of glucose joined together by a chemical bond, that is glycosidic bond, then it is polysaccharides. So you try to understand why it is known as poly, means monomeric unit are joined in a chain, and why it is mono, means single unit is present, and why it is di, means 2 unit of monosaccharides. Any monosaccharide joined together by a chemical bond, that is glycosidic bond, that is known as disaccharides. Okay, so generally we can study the chemical structure carefully and try to identify whether it is alpha form of glucose, whether it is the beta form of glucose, or whether it is a D-glucose. Why it is known as D-glucose? If you draw the structure and if you will find second last carbon in any monosaccharide, hydroxy group is present on the right side, then it is D-glucose. If hydroxy group present on the left side, then it is L-glucose. So just remember this type of terminology in chemical structure of biomolecule, especially carbohydrates. In next day, I will discuss about chemical structure of amino acids. You have to also understand the chiral carbons or achiral carbons in case of amino acid, and then you have to remember the total number of carbon in different amino acid. You know there is a standard amino acid that is coded by a specific codon, and if it is not coded by a specific codon, then it is known as non-standard. In case of a standard category, there is a essential amino acid, the amino acid which is not synthesized in your body, that is known as essential amino acid. You are taking through the diet, and some amino acid which is going to synthesize in your body, then it is known as non-essential amino acid. So try to remember, how can you remember the 10 essential amino acid? Like if you remember, treat very ill, empty at hospital. So tryptophan, threonine, valine, isoleucine, leucine, lysine, methionine, phenylalanine, arginine, histidine. So these are essential amino acids which is not going to synthesize in your body, and rest other 10 are non-essential which is going to synthesize your body. So in next class I will discuss the chemical structure of amino acid. You know protein. Protein is macromolecular, linear polymer, high molecular organic weight compound joined together by a peptide bond. There is no glycosidic bond occur in case of protein, only peptide bond. So beside peptide bond, some other bonds are present. In case of protein, like covalent bond, ionic bonds, hydrogen bonds. So we will discuss detail in next class. Thank you and have a nice day.